Vaccine and Vaccination, a primer with a focus on COVID-19, December 8, 2020. The history of vaccines and vaccinations is long and extensive, stretching back thousands of years. Vaccines have been hailed as one of the most important, if not the most important, medical advance for achieving reduced morbidity and mortality on every continent, in every country, and every community throughout the world. Nevertheless, even with the experience, know-how, and technologies available to us today, we have yet to produce vaccines for some of the most common and deadly diseases afflicting mankind. It may be pertinent to start with a very brief history of vaccines and vaccination. As with many medical achievements, vaccines and vaccinations started with keen and insightful observations. As documented by the Greek historian Thucydides more than 2,400 years ago, it has long been known that individuals who already had been sick from a disease, in this case the plague of Athens in 430 BC, and survived, were less susceptible to become ill from the same disease a second time. Although the mechanism behind this and similar observations were not recognized at the time, there was certainly an appreciation for what today is known as acquired immunity. The word immunity comes from the Latin words immunitas or immunis, both which can be understood as exemption. Techniques to stimulate the body's immune response to a pathogen have probably also been used for more than 2,000 years. Grinding up smallpox scabs and blowing the matter into the nostrils, as well as exposing matter from a smallpox to lesions on skin of a susceptible individual in order to achieve immunity, may have been employed as early as 200 BC in China. Similar attempts to prevent diseases, especially smallpox, were employed in Europe as early as the 1600s by a technique called variolation. Variolation consists of transfer of pus from the lesions of a person with active smallpox to another person, usually subcutaneously by the means of a lancet. Another word used interchangeably with variolation is inoculation. In 1718, Lady Mary Wortley Montagu had her son variolated in Constantinople by Dr. Charles Maitland. Lady Montagu, whose husband was ambassador to Turkey, had been disfigured by smallpox around 3 years earlier. She had heard about variolation upon her arrival in Turkey and was very anxious that her 6-year-old son, Edward, have the procedure. She is today given the credit of introducing this technique to Britain and the rest of Europe. The foundation for modern immunization, though, has been credited to the works of the British physician Edward Jenner. In 1796, Jenner inoculated a young farm boy, James Phipps, with pus from the lesions of a milkmaid infected with cowpox. 6 weeks later, Jenner introduced, through variolation, smallpox on 2 sites on Phipps' arm, but these and subsequent exposure could not elicit an active disease stage. Although Jenner is universally recognized for his efforts in performing and popularizing vaccination against smallpox with the help of cowpox, others had used his techniques decades earlier. The term vaccination was for the next 100 years only used when referring to cowpox inoculation to prevent smallpox. This changed when in 1885 the French chemist Louis Pasteur developed and introduced an antidote to rabies. Due to the very long incubation period of rabies, this antidote essentially functioned as a vaccine, even though vaccination originally only referred to prevention of smallpox through the use of cowpox, vacca being the Latin word for cow, while vaccinia is the Latin word for, or the Latin name for cowpox. The use of the word immunization is today used interchangeably. It's important that when we discuss topics such as vaccine and vaccination, we need to have a shared understanding of terms. On this slide, you will see stages of reduction of infectious diseases by vaccination as defined by the World Health Organization. Control means reduction of prevalence and incidence of a disease. Elimination of disease, reduction to zero, the incidence of a disease in a defined geographic area. Elimination of infection, reduction to zero the incidence of an infectious disease in a defined geographic area. Eradication, permanent reduction to zero the worldwide incidence of an infection caused by a specific agent, and one example is smallpox, where the last naturally occurring smallpox occurred in 1977. An extinction, an infectious agent no longer exists in nature or in a laboratory. Antibody. Proteins of the immunoglobulin family present on the surface of B lymphocytes secrete in response to stimulation that neutralize antigens by binding specifically to their surface. An antigen is a toxin or a foreign substance or any molecule or partial molecule that triggers or induces an immune response. The immune system is a complex network of cells and proteins that defends the body against infections. Immunity is a protection from an infectious disease. And when we talk about vaccine, we talk about a product that stimulates a person's immune system to protect the person from that disease, while vaccination immunization is the act of introducing a vaccine into the body to produce immunity to a specific disease. The success of vaccines is based on the utilization of the body's own very complex and yet remarkable immune system. An appreciation of how the immune system helps to protect us from foreign pathogens is needed to better understand the modes of action, benefits, risks, and potential real-life impact on protection by different vaccines. An immune response can broadly be divided into an innate immune response and an adaptive or acquired immune response. While the innate immune response occurs within minutes to hours, the acquired immune response takes days to appear. The innate immune response doesn't respond to any one specific antigen, but to all antigens it's been exposed to, while the acquired immune response is antigen-specific and can recognize antigens that this body has been exposed to previously. The innate immune response doesn't have any memory, while the acquired immune response actually generates memory cells, and these memory cells is the key to an effective immunization. The innate immune response utilizes several different cells such as neutrophils, eosinophils, basophils, monocytes and macrophages, mast cells, dendritic cells and natural killer cells in a response to pathogens that the body may be exposed to, while the acquired immune response mainly consists of lymphocytes that are divided into T lymphocytes and B lymphocytes. The T lymphocytes are further divided into T helper cells or CD4 positive cells that are further subdivided into T helper 1 and T helper 2 cells and T cytotoxic cells or CD8 cells. The B lymphocytes eventually differentiates into plasma cells that are responsible to produce the antibodies to protect us against the foreign pathogens that we may be exposed to. Here is a very simplified explanation how the acquired immune response might look like. A person gets infected by a virus. The virus is picked up by an antigen-presenting cell that breaks down the components of the virus and uses some of the virus components in order to stimulate lymphocytes that will differentiate into T lymphocytes and B lymphocytes. The virus can also directly stimulate B cells to be differentiated and respond. The antigen-presenting cells will further differentiate or help to differentiate the T lymphocyte into CD4 cells and CD8 cells. And furthermore, the antigen-presenting cells will also help to strengthen the actions of the CD8 positive cells to become cytotoxic T cells that will kill infected cells. The CD4 cells, as mentioned earlier, are divided into Th1 and Th2 cells. The Th1 helper cells enhances the CD8 cells to produce the cytotoxic T cells, while the Th2 helper cells promotes the B cells to generate plasma cells that eventually is going to generate protective antibodies against pathogens that we may be exposed to in the future. Now, very importantly, both the CD8 cells and the B cells will produce memory cells. Now, this is what we want to achieve through vaccinations. A virus is an organism that will try to insert its genetic material, RNA or DNA, into a host cell. The virus in itself cannot replicate and needs a host cell's mechanism or machinery in order to produce more viruses. The figure to the left shows a microscopic picture of the severe acute respiratory coronavirus 2. It looks like the virus is surrounded by a crown or a corona, which gives it its name. The right figure is an artist's now famous rendition of the virus, where it is possible to notice the protruding elements that make up the crown in the microscopic picture. The protruding elements are named spike proteins and are used by the virus to attach and subsequently enter a cell to release its genetic information. Specifically, they are part of the spike protein named receptor binding domains that are responsible for attaching the virus to a receptor, the angiotensin converting enzyme 2, an ACE2 receptor that is found on most mucosal cells throughout the body. Apart from the spike protein, the virus has other proteins such as the E or envelope protein, the M or membrane protein, and the N or the nucleocapsid protein. Although all of these proteins will elicit an antibody response, vaccine development particularly focus on the development of antibodies to the spike protein, or more precisely to its receptor binding domain, to prevent the virus from attaching to its cell. But nucleocapsid protein is also being targeted for vaccine development in the future. This cartoon depicts in a very simplified way how the SARS-CoV-2 virus attaches to the angiotensin converting enzyme receptor on a cell and subsequently releases RNA into the cell in order to utilize the cell's ability to generate proteins, in this case viral proteins, in order to produce more viruses. The cell will release the produced viruses, which will contribute to stimulate the body's immune response, as shown on previous slides. The key to vaccine development is to produce a humoral, antibody, immune response and a cellular immune response, a cytotoxic T cell, to produce long-lasting memory cells without causing an infection. In other words, an effective vaccine will trick the body's immune system into believing an actual infection has occurred. Several different schemes are used today to produce vaccines. The 5 most common ones are vaccines using weakened and attenuated viruses, inactive or kill viruses, viral genetic materials such as RNA and DNA, or subunits of the virus, or viral vectors to produce an immune response. Weakened and attenuated viruses are used for several vaccines available today. Although this technique will produce a robust immune response, generating long-lasting memory cells, weakened virus vaccines should not be used in individuals with immune deficiency, as such individuals may actually develop disease, for example measles, if a weakened measles virus is being used as a vaccine. Protein-based vaccines are a little bit different. Depending on the actual methods being used, these vaccines are called subunit, recombinant, polysaccharide, and conjugated vaccines. A subunit, a component of a microorganism such as a protein, a sugar like a polysaccharide, or the capsid of a microorganism, is used as the antigen to provoke an immune response. These types of vaccines often require adjuvants to produce a strong or stronger immune response. Vaccine adjuvants help to hasten, augment, and extend the immune response stimulated by a vaccine agent. One such adjuvant technique, sometimes described as a conjugated method, is when part of a bacterial coat is combined with a carrier protein to enhance the immune response. Once vaccinated, the immune system recognizes that the proteins don't belong in the body and begins making T lymphocytes and antibodies, and if infected in the future, the memory cells will recognize and fight off the virus. Another type of vaccine may use an antigen, a virus-like particle that is produced by a viral protein that resembles or is a subunit of the actual virus. This is one way of producing a vaccine against multiple strains of viruses, for example, human papillomavirus strains. These vaccines are used to protect against diseases and pathogens such as Haemophilus influenza type B, hepatitis B, HPV, whooping cough pertussis, pneumococcal disease, meningococcal disease, and shingles or herpes zoster. Viral vector vaccines consist of a recombinant virus, that is, the viral vector, a different virus than the one that caused COVID-19, often attenuated to reduce its pathogenicity, in which genetic material from the virus that causes COVID-19 have been cloned using recombinant DNA techniques. Vector vaccines can either be replicating or non-replicating. Using these instructions, cells make copies of the protein, in this case the spike protein. Replicating vector vaccines infect cells, and once the viral vector is inside the cell, the genetic material gives cells instructions to make a protein that's unique to the virus that causes COVID-19 in our case. Non-replicating vector vaccines initially enter cells and produce the vaccine antigen, but no new virus particles are formed. Because viral vector vaccines result in endogenous antigen production, both humoral and cellular immune responses are stimulated, and both T lymphocytes and B lymphocyte memory cells are being produced. These recombinant vector vaccines, by the way, are also being studied for use against other viruses such as Ebola, human immunodeficiency virus HIV, H5N1 avian influenza, and Zika virus. Another type of vaccine are vaccines that are based on nucleic acid. Nucleic acid-based vaccines can consist of DNA or messenger RNA and can be adapted quickly when new viruses emerge, which is why they were among the first COVID-19 vaccines to enter clinical trial. DNA plasmid vaccines produce an immune response by inserting parts of viral DNA into a human cell that will now be able to produce a viral-like protein that can elicit an immune response. DNA vaccines consist of a synthetic DNA construct encoding the vaccine antigen. For efficient uptake of the construct into the cells, injections need to be followed by something called electroporation, a method used to create pores in the cellular membranes. After uptake into the cells, the vaccine antigen is expressed from the DNA construct. The mRNA vaccine or the messenger RNA vaccines contain materials from the virus that causes COVID-19 that gives our cells instructions for how to make a harmless protein that's unique to the virus. By inserting parts of viral RNA into a human cell, the cell will now be able to produce a viral-like protein that can elicit an immune response. After our cells make copies of the protein, they destroy the genetic material from the vaccine. Our bodies recognize that the protein should not be there and produce T lymphocyte and B lymphocyte memory cells that can be activated in case of any future infection. One of the first vaccines that will most probably get authorization from FDA or the Food and Drug Administration is the Pfizer vaccine. Pfizer vaccine is based on the messenger RNA platform. This slide here was produced by Pfizer in order to explain how their vaccine works. The genetic sequence of the spike protein is used to synthesize a messenger RNA sequence. The synthetic messenger RNA is packed in a lipid nanoparticle, LNP, that serves as the delivery vehicle to a cell. Once inside the cell, the cellular machinery follows the messenger RNA instructions to produce the viral protein, which is then displayed on the surface of the cell and stimulates the immune system to mount a response. Many of you might have heard that this type of vaccine needs to be kept at a very, very low temperature, and the reason for that is actually to protect the lipid nanoparticle for not breaking up too early. Experts today contend that there are several factors that argue for the messenger RNA vaccine's safety. For one, messenger RNA can't cause an infection. It also doesn't enter the cell's nucleus, so the chance of its integration into human DNA is believed to be very low. In addition, the body breaks down the messenger RNA and its lipid carrier within a matter of hours, which will lessen some concerns about long-term risks. However, this rapid degradation raises questions about messenger RNA vaccine's protective duration. Vaccine development goes through different phases before being potentially authorized or approved by, in the US, the Food and Drug Administration or FDA. It starts with preclinical testing in laboratories and animal testing. Phase 1 safety trial tests for safety, dosages, and immune response in a small number of people, usually healthy people. Phase 2 safety trial tests for safety, dosages, and immune response in a larger group that consists of different cohorts, for instance children and elderly. And then phase 3 safety trial is when the vaccine and a placebo are given to thousands of people to ascertain the vaccine efficacy. FDA requires at least 50% efficacy in order to authorize or approve a vaccine today. Today there are at least 86 vaccine candidates going through clinical testing, and an additional 58 vaccines are used in clinical trials. Many of these vaccines come from different companies. I have listed some of them here: Moderna, Pfizer. There are a Chinese company, CanSino Bio, the Russian vaccine Sputnik, Johnson & Johnson or the Janssen vaccine, AstraZeneca, Oxford, Novavax, Medical Cargo, another Chinese vaccine from the Institute of Medical Biology in China, and the Sanofi vaccine from GSK, which is a protein vaccine, and the same company is actually now also working on an mRNA vaccine. Moderna, Pfizer, Johnson & Johnson, AstraZeneca, Novavax, and Sanofi will probably be the 6 primary candidates for FDA authorization, and those are probably the vaccines that you're going to see in the near future. Important to know is that the Moderna and Pfizer, both of them are genetic vaccines, mRNA vaccines. The Johnson & Johnson or the Janssen vaccine and AstraZeneca use vectors, adenovirus, human adenovirus, as well as chimpanzee adenoviruses. Novavax and Sanofi are subunit or protein type of vaccines. Could there be other types of vaccines? As shown with the polio vaccine, eliciting the mucosal immunity may be another effective approach to achieve an effective immune response against SARS-CoV-2. The rationale for mucosal vaccine is very clear, as oral or nasal mucosal surfaces serve as the primary portal of entry for pathogens like coronaviruses in the human body. As evidenced by studies on the similar coronaviruses, SARS-CoV-1 and MERS, Middle Eastern Respiratory Syndrome coronavirus, mucosal vaccination can provide a safe and effective means for induction of long-lasting systemic and mucosal immunity to confer protection against SARS-CoV-2, which makes a lot of sense considering that the receptor for SARS-CoV-2, the ACE2 receptor, is found on mainly on mucosal cells throughout the body. When reading manufacturers' report on vaccine efficacy, we have to make a differentiation between if the vaccine could prevent an infection or if the vaccine actually reduced disease. There is a big difference. Being able to reduce an infection means that we don't have any replication of the virus in the body. If we don't have an infection, it cannot cause disease. And if we don't have an infection, we cannot transmit the virus. On the other hand, if the manufacturer report that their vaccines reduce disease, meaning less people develop disease if they receive the vaccine versus receiving the placebo, it doesn't say anything about if it truly reduces infection, i.e., did it actually prevent the virus from replicating in the body? Did it really prevent infection? Vaccine efficacy is measured by the relative reduction in infection or disease, depending if we are looking at infection or disease, of the vaccinated cohort, the intervention cohort, versus the unvaccinated cohort. It's a relative risk that we are looking at. For example, vaccine efficacy of 75% roughly means that you have a 75% reduced risk of becoming sick compared to an otherwise similar unvaccinated person. Most phase 3 trials are measuring efficacy to prevent disease as the primary analysis, with efficacy against severe disease and against infection as secondary aim. Preventing infection entirely is the hardest to achieve. A vaccine that can prevent infection will also prevent disease and severe disease, but we can have vaccines where people are still infected but their disease severity is lessened. A vaccine that prevents infection entirely provides indirect protection to others. If one cannot get infected, one cannot infect someone else. But it is possible to have a vaccine that prevents disease, but the individual can still be infectious. How well a vaccine directly protects the vaccinated individual is one aim. Another aim is for the vaccine to reduce infectiousness to others. This is known as indirect protection and is related to herd immunity. A form of indirect protection from infectious diseases occurs when a large percentage of the population has become immune to an infection, whether through previous infections or through vaccination. And it provides a mean or a measure of protection for individuals who are not immune. You have on the left side here a group of individuals. Most of them are not immune and still healthy, and you have 2 individuals here that are contagious and sick. Obviously, over time, as no one is really immune, you're going to have a lot of the not immune and previously healthy people getting infected, and then contagion spread throughout the population. Another situation is where we have non-immune healthy people, sick and contagious people, and some immune and healthy individuals. Over time, we are still going to get contagion and spread throughout some of the population, maybe a little bit less than previously, but there's still going to be a significant amount of people that are going to be infected. However, if we have a lot of people in the community that are immune and healthy, over time this is going to prevent the spread throughout the population. Over time, this is going to result in less spread of disease within the population, and this is called herd immunity. It estimated that for COVID-19, we need a minimum of 70% of the population to be immune, either through natural immunization or through vaccination, in order to achieve this type of herd immunity. Now, what are the outstanding issues that we need to look at when these vaccines are being produced or being put on the market? Number one, do we know the duration of protection through this vaccine? And that is not clear yet. We don't know exactly when we need or how many booster doses we're going to have. We see in some of these vaccines coming out that some individuals will actually develop some type of immune response after one dose, but they will boost their immune response after 2 doses. Will that be enough in all subpopulations that are going to get this vaccine? So the effectiveness in subpopulation has really not been evaluated to the full extent. Will the vaccine be different in a child, in an elderly person with multiple morbidities, and so on and so forth. And obviously we don't know in the end the impact on community transmission, and the reason for that is that these vaccines are being very, very quickly authorized and approved and have only been tested in a small number of people, up to around 30 to 40,000 individuals. In order to see the impact on community transmission, we need to have this vaccine in the community for a longer period of time, so we can actually see the effect of having many more than the 30, 40,000 people being vaccinated. This is an excellent review article from the Clinical Microbiology Review published by the American Society of Microbiology in 2019. In this article, the authors actually address 2 issues. One is they get very much into detail of the methods for quantifying vaccine responses. So it's not only seeing how many people get the disease or not get the disease, or see if they develop antibodies. The question is, how robust are these responses? They're looking at the innate responses, natural killer cells, dendritic cells, monocytes, and so on and so forth. They look at humoral responses, look at different type of immunoglobulins and how well they work. They look at cellular responses, obviously cytotoxic T cells and others, and how well they work. Cytokine responses, and there are future methods that involve gene expression profiling and epigenetics and microbiome analysis. If you want to know more about it, please go to this article. Again, it's excellent, as well as the topic of factors to influence the immune response to vaccination. And obviously these factors are the ones going to determine how effective a vaccine is going to be. If we now test the vaccine in phase 3 trial, can we actually have all the subgroups of these factors involved in the test? And the answer is absolutely not, because it would be too large of a trial. But intrinsic host factors such as age, sex, genetics, comorbidity are clearly factors that can affect the efficacy of the vaccine, and we need to have the vaccine trials in phase 3 looking at both the age, sex and gender, genetics and comorbidity. There are perinatal host factors, extrinsic factors, behavioral factors, nutritional factors, environmental factors, vaccine factors, and administration factors, the scheduling and where you give the vaccine, the route of the vaccine, the time of the day even, or co-administered vaccine with other vaccine and other drugs. I think this is very important information if you want to know more about vaccines and how they work. Another tremendous resource is Center for Disease Control and Prevention. They have updates all the time on different type of vaccines and how they work. Very importantly, also on the CDC website, you will have the Advisory Committee on Immunization Practices publishing their findings as well. Now, the immunization schedules are several, obviously, and I'm not going to go into those into details here. You can go into the CDC website to find them. There are recommended adult immunization schedules, the recommended adult immunization schedule for people with medical conditions, for children and adolescents, catch-up immunizations, and so on and so forth. These are being updated on a regular basis, obviously, so please go into the CDC website where you'll be able to find all of these immunization schedules. I often get questions about flu vaccinations and COVID. Is this something we should do? Should we take the flu vaccine today when COVID is around? And I think CDC in their MMWR report from August 21st this year came out with an answer that we should pay attention to. The 2020-21 influenza season will coincide with the continued or recurrent circulation of SARS-CoV-2. Influenza vaccination of persons aged 6 months and above to reduce prevalence of illness caused by influenza will reduce symptoms that might be confused with those of COVID-19. And this is obviously extremely important in order to reduce the amount of people that needs to be seen in the hospital and maybe being treated and take up the place for people who have COVID-19 who truly need to access hospital services today. The role of healthcare professional immunizations are several. We need to be knowledgeable about the different type of vaccines and even immunization tables, and very importantly adverse events to different vaccines. We also need to be knowledgeable about how to educate patients about vaccines, how to advocate for immunization, and in case we want to perform immunization, we need to be able to educate and train ourselves in order to do it appropriately, which includes, by the way, very importantly, reporting requirements that may differ from state to state and country to country. I hope this short presentation addressed some of the questions you have about vaccine and vaccinations. Obviously, I couldn't go into more details about all the different type of topics that might interest you, but hopefully we might be able to do that in future presentations. So thank you for your attention, and please stay safe.